Hello and welcome to another section of Molecules of Life. This time we will not just be looking at molecules and elements, but we will be looking at the large macromolecules that make up all the structure of life. Again, as part of this class, we're kind of looking at the scientific method, and what we want to do is to tell you is that a large proportion of our society is actually sensitive to lactose. Lactose is a disaccharide that's present in sugar, and that individuals, and so here's a bonbon which has this kind of lactose molecule. Here's one sugar, and here's the other sugar. These sugars are not basically digestible by people once you reach maturity. Again, the only people who really need, I guess, evolutionary, who need to break down lactose are babies that eat milk. And once you get older, you basically don't have this anymore. What's really kind of interesting is that we have a whole set of people, basically Northern Europeans and certain African tribes that consist milk, and they have the genes for this. And so that's kind of really interesting. And so that's where evolution comes into us. And that, again, we've evolved to use lactose as children, and we've evolved to use lactose, and certain societies have evolved to use lactose to get the most nutrients out of their environment. Today we're going to talk about organic compounds, which are basically carbon-based molecules. These are the majority of the molecules by weight that make up our body. Again, like we said before, carbon is a very versatile atom because it has 4 valence electrons, and those outer valence can hold up to 8. So that means that carbon can produce kind of 4 individual bonds to others. And so here we look at it. And so here's the carbon skeleton that we have here. Here's various carbon skeletons, and carbons can actually also form rings as well. And so these are very versatile structures. Again, we can also have double bonds. So this carbon has a 1, 2, 3, 4 bonds on there. I'm not going to get into that much because we're going to leave this for our chemistry class. But the simplest organic compounds are what we call hydrocarbons. It's like carbon that is attached to basically water, H2O. Again, when we look at these carbon molecules, we can look at different structures. Here's the structureless stick formula that we will use a lot. Here's a ball and stick model that you will make in chemistry once you take that. And this is what we call a space-filling model. This model is very interesting because it gives us the true three-dimensional structure. This one here is easiest to understand. This one here is what we need to look at in order to see what actually goes on in space. Okay. Again, the larger hybrid compound goes for is fuel for engines. So like a gasoline is typically a benzene ring. Fats that we see here, this is fatty acids that are attached to a glycerin over here, gives us the structure in here, and we'll talk a lot more about what they are. And again, the structure function tends to show up a lot here is because these molecules have a site to them, a size to them, and it's this size that gives them their reactivity. The second thing I want to talk about is attached to the carbon molecules can be what we call these functional groups that give it its activities. And so again, the carbon backbone, the carbon backbone is what gives the overall structure of a molecule, but it's these functional groups that give it its activities, and it can allow that molecule to be soluble in water, hydrophilic, or not soluble in water, hydrophobic. The main macromolecules that we're going to talk about are carbohydrates, proteins, and nucleic acids. Your book does not include fatty acids in there because those molecules tend to not be as large, but I would counter that, yes, they are pretty large, and they're very darn important too. So let's put in fats here, and we'll talk about those. Briefly, what I want you to get out of today's lecture is that what these molecules look like and basically what these molecules will do. So we'll go over that real quick. Again, we talked about it before, but these molecules are what we call polymers. Polymers are basically repeating units of what we call monomers. The monomer is the smallest structural unit of this, and polymer is what it's made up of. Imagine a brick wall is a polymer of individual bricks. Each individual brick is the same, but you put those bricks together and you have a wall. Or let's say pearls in a pearl necklace. Each individual pearl is a monomer, and the pearl necklace together is what we call a polymer. This occurs by what we call a dehydration reaction, and we'll try and talk about this a little bit later on in the class. But in this dehydration reaction, this is how we put monomers together and how we take monomers apart. So it's very critical that you kind of have an awareness of what's going on here. And so here we have a short polymer. When we take that short polymer and we add it to a monomer, we actually typically remove a hydroxyl group from the short polymer and a hydrogen from the monomer, releasing water. And then so when we make this larger polymer, we're releasing water. So this is one of the things that organisms can use to actually make water when they don't have water in there. Lastly, water is very important because in order to break these polymers apart in a process that we call hydrolysis, hydro meaning water, lysis meaning to break apart, we will add a hydroxyl group to one part of the polymer and we will add a hydrogen to the other part of the polymer, and that will safely break these chains. Again, know this, hydrolysis is where we break the bonds between monomers. It adds a molecule of water. It reverses the dehydration reaction. And so again, the four categories of large molecules that we're going to go through real quickly here are carbohydrates, lipids, proteins, and nucleic acids. And we will talk about these. Carbohydrates are the sugar polymers. They include the small sugars that make our soft drink sweets. They include the large starch molecules that we see in pasta and potatoes, as well as the cellulose that makes up trees. Monosaccharides are what we call mono, meaning one. Saccharide is basically sugar, are simple sugars that cannot be broken down by hydrolysis into smaller sugars. Again, the enzymatic reactions of your body can break them down into carbon dioxide, but that's something that we'll leave until a little bit later. Glucose and fructose are what we call isomers. These are molecules that have the same molecular formula, but they have different structures. Again, you may say, well, if they're both 6-carbon molecules, but you know what really is important here is that because they have different shapes, they have different characteristics. Believe it or not, fructose is actually sweeter than glucose. So if you like Mexican Pepsi that has glucose inside of it, it's actually not going to be as sweet as the one that has the high fructose corn syrup that we all know and love. Again, this is looking at the structure of glucose. This is looking at the structure of fructose. As we can see, this unit over here is aldehyde, and the ketone is slightly different. And it's this different that our body can sense. And believe it or not, this one here is a little bit sweeter than this one over here. And so if you are a provider of sugar to any company, you would rather sell fructose than you would sell glucose. Okay, and we'll talk about that a little bit later. This is looking at the overall structure of what a 6-carbon glucose molecule would look like. We can represent this as a linear ring and as a circle. This is the abbreviated structure with the hydrogen ions and the hydroxyl groups not on there. And basically, chemists would tell you that kind of both of these exist in the equilibrium and that they are constantly kind of switching back and forth. A disaccharide is a double sugar constructed of 2 monosaccharides. It's the simplest polymer that we have out there. And believe it or not, we have 2 types of 6-carbon sugars. These are alleles of one another, not alleles, isomers of one another. We have glucose and galactose. Glucose and galactose together make up lactose, which is basically milk sugar. And again, there's a fair number of you who are lactose intolerant. If we increase the size of that sugar, we have maltose, which we see on malted milk, malted barley. Sucrose is a table sugar. Sucralose is a alternate form of it that's not digestible by anyone, even bacteria, so that even though it tastes sweet, you can't see it. And we're going to kind of move on from there. What's really interesting is to look at this process in biotechnology. This is very important because basically we could take something like starch from corn. We break down this starch into glucose. This is a sweetener, but then this glucose is converted into fructose, high fructose corn syrup, because it's quite a bit sweeter. And so again, you don't have to add as much. And so Coke companies can add a little bit less inside of there. And again, for the most part, there's no really difference. There's nothing really bad about high fructose corn syrup, except that we drink too much of it. and even sugar, even honey, which is a fructose concentration, is bad when you eat too much of it. Again, in the United States, we typically consume about 45 kilograms or about 100 pounds of a year. That's kind of way too much. Polysaccharides are complex carbohydrates. They're made of long chains of sugar units and polymers of monosaccharides. The glucose polymer, you see here, and then the cellulose polymer. What's really kind of interesting is the starch polymer. If we look at the starch polymer, it exists as a helix in here, and water molecules can get in here pretty easy. These cellulose, basically these molecules actually have cross, if you notice, these don't attach to one another, and these do, and so the starch molecules are much harder to kind of get rid of. Starch is an example of polysaccharide that's used by plants to store energy. Potatoes and grains are major sources of that. Glycogen is basically starch used by animal cells. There's a little bit more cross reproducers. Cellulose is the most abundant compound on Earth, based because there's not only is there a lot of it made, but it's really tough to kind of break down. All animals cannot break this down. The animals that can, like cows, etc., have symbiotic relationships that allow them to do it. Okay. The next polymer that we want to talk about are lipids. Lipids are different than almost all the other ones in that they are hydrophobic. They don't necessarily like water. The reason is because they don't have those active units that will actually bind with water, that will hydrogen bond with water. And so since they don't hydrogen bond with water, and water likes to bind with itself, it excludes it. And so, again, when we take water and we put it in an aqueous solution, so when we take oil and put it in an aqueous solution, that water is going to separate out. Okay. I do want you to have a general idea of kind of what fats look like. Again, we have this long chain with nothing on it. So this is a nonpolar molecule. This is a fatty acid, and this is the acetyl group, carboxylic acid group that's on side of there. And then this is the dehydration reaction that occurs to attach a fatty acid onto a glycerol. Fats are used for energy storage, for cushioning, and for insulation. Two things that we want to see is what we call unsaturated fats and saturated fatty acids. Hydrogenization is where we basically randomly add a hydrogen to it. This will create some trans fats, which basically are not good for you because the body does not know how to kind of break down fats. Saturated fats, again, include things like your meats and your butters and stuff like that. And these are molecules that basically need lower temperatures to be solids. The unsaturated fats are a little bit more kind of liquidy, and we tend to see them more in plant oils and omega-3s. And so we'll kind of go on from there. Steroids are very different. Steroids are based on the cholesterol molecule, which is this kind of flat planar 1, 2, 3, 4 circular rings. And again, it's very little difference. This difference right here is what can sterol that would indicate maleness, and a sterol that would kind of signal femaleness. And so we'll talk a little bit about this a little bit later. But again, synthetic anabolic steroids resemble testosterone. They also mimic its effects. They can cause serious physical and mental problems. Again, the normal steroids in high concentrations can do this as well. And these are some of the earlier drugs that were abused by athletes. And I'm not going to kind of get into that that much. Proteins, again, are very important because these are polymers constructed from amino acid monomers. They perform most of the tasks that the body needs to function. They form enzymes, chemical, and change the rate of chemical reactions without changing the process, and that's called a catalyst. We have structural proteins, which include the hair and horns and nails. Storage proteins that we tend to see a lot in plants and eggs. We have contractile proteins, our muscles. We have transport proteins in our blood. We have enzymes that kind of do various things for us, and we'll talk a little bit more about that. Proteins are polymers of these things that we call amino acids. There's 20 amino acids that we have naturally inside the body. And the thing about the amino acids is this structure right here is they have an amino group, hence the amino part of that. They have a carboxylic acid group, hence amino acid group. These are attached to an alpha carbon, and it's the side chain that gives the property of this. And so here's an example of that amino acid kind of alpha unit, and then this properties give us leucine. Here's one amino acid. Here's serine. Here's the other amino acid. They give us the different properties that are on there. Okay, and so that's we'll talk a little bit more about that later. Amino acids are held together by a peptide bond, and this is the dehydration synthesis, right? So when we remove that water from there, we get left what's called a peptide bond, and we get amino acid registers. Now, proteins are very important because they have a very interesting three-dimensional structure. And again, how proteins get this three-dimensional structure is we have this specific primary structure, which is the amino acids together. Those amino acids can fold together to give us another structure. Amino acids are very important. It's one change in one amino acid, this leucine, glutamine, to a valine that changes our red blood cells from this normal donut shape to the sickle cell, which this is the disease state that we have in here. And again, there's a number of reasons for that, and I'll talk about that. The protein shape has three levels. We have what's called the primary level, we have the secondary level, we have the tertiary level, and the quaternary structure of proteins. Protein's three-dimensional shape gives basically the structure of the molecule. And what we're going to look at here is kind of how this works. And so here's an enzyme, here's the protein, here's the target molecule. And what's really kind of interesting is that target molecule will fit directly into pockets in here, and that pocket can be altered and strained, and we will talk a little bit about that later. Denaturation is where basically pH or temperature changes can change and unravel a protein. High fevers above 140 degrees. 140 degrees are very bad in human beings because they cause our proteins, especially the one in our brains, to start to denature. Once they denature, basically we're dead. Another problems with denaturation that we have are things like Alzheimer's disease, mad cow disease, and Parkinson's disease. The last molecule that I really want to talk about are nucleic acids. And we talked a little bit about this in class on Monday, where we looked at this. Nucleic acids we really understand a lot because, again, they're relatively simple molecules, but they're simple, but they're not simple at the same time. And what we really need to know is that a gene is a specific stretch of DNA that programs an amino acid sequence of a polypeptide. This is a really simple definition on here, but for right now, that will go. DNA is the nucleic acid language, protein, and then your amino acids make up the protein language, and we'll talk about that a little bit later. So here's a gene. That gene is converted into RNA, and that RNA is then used to produce protein. We will have a whole chapter on this a little bit later. The nucleotide, so nucleotides consist of a phosphorus group, a sugar backbone, and this is deoxyribose sugar, and then here's thiamine on here, a nitrogenous base, and that's the A's, G's, T's, or C's that we're talking about. And here's a little cartoon of kind of what that may look like. Okay, again, know these and know their base pairing. This is a space filling model of what a DNA molecule would look like. Here's what the individual base pairs look like, and what they do is they kind of bind together. T's always bind with A's, C's always bind with G's, and they have kind of different sizes on here. But again, when they're together, this gives us a DNA molecule that has a particular shape. It's almost rod-shaped, that molecule. A's only paired with T's, and G's only paired with C's. This gives us the double helix. RNA is slightly different. It's actually a ribose sugar because it's not deoxyribose. It actually is ribose because it has that sugar. And then I'm not going to kind of get into this, but most lactose intolerant people are actually actually have a change in their genetic sequence, not in the lactose genes, but above it. And so what's happening is this individual, before he got his beard when he was a little baby, was able to break down lactose. As an adult, he doesn't need to break down lactose, so he kind of lost that particular gene Here's one of the early cows, domesticated cows that we have. Here's the dehydration synthesis. This is a very important structure to look at because it allows us to look at kind of what carbohydrates, lipids, and proteins are. We look at their functions. You need to know their components, and you need to give examples of these. And hopefully we'll do this in class a little bit. Okay.